in very simple terms, our brain cells, also called neurons, they communicate with each other using electrical signals. And to create and control these signals, they use tiny molecular little gates, and they are called ion channels. And you can imagine them like little doors in the cell membrane in our brain that open and close and let those ions pass through. And now if there is a mutation in one of these genes that builds such a channel or little gate and may not work properly, and that can lead to seizures or other neurological symptoms.
Hi, everyone. I'm Dr. Nancy Musarra, and you are listening to the KCNA2 and Rare Epilepsy Podcast. Hi, everyone, and welcome to the KCNA2 and other Rare Epilepsy Podcast. Today is one of our very special episodes because today we're going to be talking to a real researcher who researches rare epilepsy. And what we'll get from today is answers to the many questions parents have once they get a diagnosis. And actually, the questions that parents have, many parents that have had a diagnosis for years still have questions about how is this rare epilepsy researched? What kind of rare epilepsies are we talking about? Where are we in terms of research? And what do some of the terms mean that we become familiar with, but we don't quite understand? So today I would like to introduce Dr. Uli Hedrich from Germany. And I know it's a 6-hour difference, but you're still awake over there in Germany. It's 12 noon Eastern Standard Time. So welcome to our show.
Thanks a lot, Nancy. And you can call me Uli. That's perfectly okay.
Okay. Uli, can you first just start out by telling us a little bit about yourself?
Sure. So I'm a junior group leader at the Hertie Institute for Clinical Brain Research here in Tübingen in Germany. I live here with my family, with my husband and my 2 little kids. And actually, I can tell you a little bit about my academic journey. So I actually started in Ulm, which is also in Germany, not that far away. I studied there biology, and later I did my PhD. And I often joke that I worked in cancer research, but actually I was working in neurobiology in that field. But I was working with cancer crabs. But this work gave me a lot of insight into the nervous system, into new techniques. And when I finished my PhD, I wanted to take these techniques and the knowledge that I gained, especially those related to neurophysiology and also cellular mechanisms. And therefore, I went into the human health. So I went into the world of epilepsy research, which really fascinated me. And today I have my focus here in Tübingen on understanding how neuronal networks become hyperexcitable, for example, during a seizure. And in addition, we are also interested in, for example, other neurological diseases like migraine and also ataxia, which are also closely related to epilepsy.
Wow. So how long have you been in your position now doing actual research on human epilepsy and neurological conditions?
So actually, I'm working in that field since now around 16 or 17 years already.
Wow.
Yeah. And
Wow. Do you find that when you're working in the field of this type of research that the pool of researchers is pretty small and everybody gets to know each other?
yes, definitely. So I also now when I go to and visit conferences, I also meet very often friends that I still know from my PhD time when I worked on a completely different system. So the neuroscientific world is also small and people know each other.
And when you meet other researchers, are there also clinicians at those conferences that you network with that actually treat patients?
Sure, of course. We are also working closely together with the clinicians And we also meet them at work, at meetings, and discuss things, talk about patients, about new possibilities.
Yeah, I ask that because so many times I hear parents, they get a diagnosis of a rare epilepsy. It can be any one of them. And they go to their neurologist, and their neurologist really knows very little about that particular variant or mutation that's been diagnosed. So it's good to know that there is a group of people that your neurologist can access that are familiar with different kinds of rare, epilepsies. So okay, so what my experience is in this field, and I've been connected to our group, which is KCNA2, but we've also connected with other rare epilepsy groups. And in all those groups, I hear a familiar story. And that story is, parents have a baby, everybody's happy, everybody's so thrilled to have this child. And then the parents start to notice that something's not quite right. It could be a developmental delay. It can be just like floppy and the muscular system seems to not be working, or they could actually see seizures already and say, whoa, we need to go to the ER. Something's not right. Once they do that, they can go to their pediatrician and possibly a neurologist, but eventually they get genetic testing. And nowadays there's more available answers in the genetic test than there was years ago, where you'd get your genetic test, but you'd wait. For my family, we waited 12 years to get a diagnosis, but now there's more answers in there. But once you get a diagnosis and you're told, okay, your child's been diagnosed with some type of channelopathy, what does that mean?
Yeah, and that's a very good question. And I think it's also important to explain what we actually mean when we say channelopathy. So, in very simple terms, our brain cells, also called neurons, they communicate with each other using electrical signals. And to create and control these signals, they use tiny molecular little gates, and they are called ion channels. And you can imagine them like little doors in the cell membrane in our brain that open and close and let those ions pass through. So such as sodium and potassium, and they flow then in and out of our cells. And this flow of ions is what also allows neurons to send their electrical impulses. So these channels need to open and close at exactly the right time and also in the right way. And now if there is a mutation in one of these genes that builds such a channel or little gate and may not work properly, maybe it's open too much, or maybe it doesn't close that well, or it's just open too long and doesn't shut down when it should. So when that happens, the electrical balance in the brain can be disturbed, and that can lead to seizures or other neurological symptoms. And this then in the end we call a channelopathy, because these little gates are altered in the disease.
So the gate is like the channel.
Exactly.
Okay. And I don't know if this is correct, but when you were saying some of the gates open, not open properly or not closed properly, or sometimes they're just not regulated, I connect that with what a lot of parents have been familiar with, which is when you get your diagnosis, you have a certain type. Some of it's called gain of function, loss of function, and mix of function. Is that what you were referring to when you were talking about gates opening, closing?
Exactly. So there are possibilities that, for example, a gate is open for, or this channel is open for a longer time. So more ions pass through that channel that than it should be. So this would be called a gain of function. And so these ion channels have to be very precisely balanced. So they open and close at just the right times to let ions flow through, and also create these electrical signals that I've told you before in those neurons to communicate. And if a mutation, for example, causes a gain of function, that means, as I said before, that the channel might open too easily or stay open for too long or let too many ions pass through. And when that happens, the neuron become, for example, too excitable. So it fires electrical signals more often, or also when it shouldn't. And when many neurons start behaving like that at the same time, the brain's normal rhythm is disturbed. And this can then in the end lead to seizures. So the same is true for loss of function. That can just mean that the channel is less active. So less ions can pass through, but this can also bring everything out of the rhythm, so of the normal rhythm, of a healthy rhythm, and can lead to seizures. So it's like an orchestra. If
Yeah.
there is if the instruments are too loud, for example, there is too much, then it's not, you don't hear the music that you would like to hear. So it's just like too much, and like it can end in a seizure.
Right. So, okay, but like I know for KCNA2 and KCNQ2, those two mutations happen to be on the potassium channel.
Mm-hmm.
There are other channels too, sodium, calcium. How different are those? Like if I have a sodium channel problem and you have a potassium channel problem, I know sometimes two different channels are more similar than two potassium channels. So how similar and different are they?
So these ion channels have actually different roles. So they are all needed to build these signals. They are called action potentials, with which our neurons, the nerve cells, communicate with each other. And for example, a potassium channel, or let's start with the sodium channel. For example, the sodium channels, they are really necessary to start this signal. And so they open, let the sodium ions flow inside the cells, which actually starts the signal, the communication. And the potassium channels, they are actually there for dampening down this a little bit, to get them back to a normal level. And therefore, both of these types of channels are so important, because you need both to keep this balance between excitation and inhibition in our brain. And if something is disturbed there, this can, of course, also result in a different behavior of our brain, and in worst case, for example, in a seizure.
That makes sense. I didn't know that's how they communicate. Like they really do work together. They're
They
not as
do.
separate as it sounds, you know? Okay, so when you are then prescribed a medication you're really targeting the symptoms of decreasing seizures, correct? How does medication affect the channel? Or does it?
So it depends. There are very specific medications that really affect different types of channels. There are, for example, sodium channel blockers, which are also often used for epilepsy patients, because they block these specific sodium channels, and they can calm down the brain a little bit. Because when you give that medication to a patient and you block this beginning of this signal that that our cells use for communication, then you can calm down the brain a little bit. But there are also, for example, potassium channel blockers, because we not only have excitatory neurons in our brain, we also have cells that inhibit other brain cells to keep that balance. And for example, if you would use a potassium channel blocker, that could also result in a different behavior of that cells, and cause then a balance, or can bring this balance back.
So when you're researching this, do you I know there's AI that you can see how a medication or an intervention affects the channel. How do you experiment and try to discover how the channel is affected by different interventions?
So we actually start quite simple. We use cells that are not really that these are not nerve cells like in our brain. These are very simple cells. They can be made out of taken out of the kidney, for example. There are cell lines available, human embryonic kidney cells, for example, which are good model cells. Or you could even use frog eggs to do some simple experiments just to check what a mutation does on a specific channel. So you can introduce those channels into those cells and then do electrophysiological recordings and see how this mutation affects the channel. How does it, for example, alter the opening or the closing of this channel? Does this channel then, due to the mutation, allow us to let more ions flow inside the cell or outside the cell? And we use these simple model systems as a first detection to get to know what a specific mutation does. So this is the first start. And then you can also go further if you would like to understand what a mutation does with the whole brain. Then you, of course, need cells that are nervous cells. And therefore, you can also use model systems, animal models, for example, rodents like mice.
Mm-hmm.
And you can bring these variants into the mouse and then do also recordings. So analyze the specific types of neurons inside the brain by using brain slices and just measure the activity of the cells if there are any differences between a healthy brain and between a brain which is carrying this mutation. But you can, of course, also work in the whole animal. So this is called in vivo. You can do also EEG recordings. You can also treat the animals in the end with a specific medication and test if this was probably would work also in a human patient. And that's the last step before you could give a medication, for example, to a patient.
So if you test something on a frog egg and it shows a good effect, that the channel is correcting or working, functioning either properly or better, does that translate to how the mice respond as well, or it could be different in mice?
It could also be different in mice, but most of the time, so in our hands, with specific blockers or activators that we very often use in our lab, the chance is very good that it also works in mice. So, but it could, of course, be also different because then you're looking on a whole network where different cells communicate with each other. And of course, there could also be a difference than by just testing it on a single ion channel.
So when you do your research, what do you like, or what are you researching, or what have you researched that has really made it like, wow, I'm so glad that I'm studying this because I'm finding out all these new things. What has been exciting for you in your research?
So I have to say, so I'm now already working since several years on KCNA2, but that was really fascinating. So I think it was in 2013 when collaborators from Leipzig reached out and asked if we could do analysis of these KCNA2 variants that they found, that they identified in patients. And when I did the first recordings in these frog eggs, and for these gain of function variants, it didn't really make any sense for us. So I first thought that I did something wrong, because I couldn't believe that this gain of function variant could lead to an epilepsy, because it doesn't really make sense. So we still don't understand how this could really result in seizures in the patients. But when I saw that in the frogs, in these frog eggs, I was really puzzled. And then when we came up with the idea that we could use this as a relatively specific blocker for this type of channel, and we tested it also in the frog eggs, and it just worked. And then the first patients were treated in the clinics, and it also worked. This was really fascinating for me. So this translation from the lab in the clinic.
What medication was that?
This was 4-aminopyridine.
Okay. Or 4-AP as we
4-AP, exactly.
refer to it.
In short.
And so when you say 4-AP worked, what worked?
So so when you have this gain of function variance in these KCNA2, in the KV1.2 channel, it's called, that means that these channels, they allow a lot of ions to flow through this little pores in our cells. And when you add 4-aminopyridine, so 4-AP, it blocks. So the current, the channels let less ions pass through. So the currents that we can record in the lab, they are getting smaller. You need a specific voltage to open these little doors, these gates. And for these gain of function variants, these voltages are much more negative than for healthy channels. And when you add 4-aminopyridine, this voltage that you need to open the channel is also shifted in the direction to the healthy voltage that you need. So it's not just blocking, it's also shifting, which is doing this positive effect also in the patients.
Now with 4-AP, it's blocking and shifting, just like you described, but is it just targeting the brain cells that are affected by KCNA2, or does it block and shift all potassium channels like in the rest of the body that are not really affected by KCNA2?
So it's not just a specific blocker for this KV1.2 channel, it also blocks other channels. And of course, if you take the drug, it blocks all the channels in the whole body. And therefore you also have to be aware that there could be also some side effects. And usually if clinicians start this treatment, the patients are in the clinic and are monitored that there are no side effects at the beginning.
So the side effects would be, although we're trying to have an effect on the KV1.2 channel, it could be affecting other channels and cause a negative result, negative symptoms or things that you don't want to see.
Exactly.
If you go to the hospital and you're titrated up carefully on 4-AP and it seems to be working and you're not getting side effects, does that mean the other channels are pretty safe, even because you can't see side effects?
I mean, they are not pretty safe. You will also, so the medication also works on them, but your body is getting along with that. So
okay, so there's it's tolerating it.
It's tolerated, exactly.
Okay. Do you think that in the future with research, somebody will try to get a more targeted or more specific 4-AP, but like a different form of it so that it only targets the KV1.2 and not the other cells? Or is that something that's so far down the road we won't see it in our lifetime?
I mean, it could be that people will develop that. There are actually also already toxins from specific animals that specifically block different types of channels. So there are already toxins from scorpions, for example, or from mollusks that specifically block the KV1.2 channel, but you don't want to give a patient a toxin. So, I mean, I can't say if somebody will develop a drug which will just selectively block KV1.2 channels. But of course, it's always the goal to have very specific drugs to get rid of side effects. But there are also other possibilities now. So there is also a lot going on in gene therapies, which could also be the future. So there is also a lot of things going on there.
You've also worked with other channels. I mean, yeah, other than KCNA2, what other research have you done?
So we are also working a lot on Dravet syndrome. That's also a very severe and rare epilepsy syndrome. And the patients are really also very severely affected. And in Dravet syndrome, these patients have variants in the SCN118. This is a gene that encodes for a sodium channel, the NAV 1.1 channel. And we are also working on that to also find new treatments, to find out what is going on there also during development. Also try to identify new mechanisms to also get new targeted treatments for this also very severe disease.
Is there a medication that people can take for Dravet syndrome, or is that still in the
There is
infancy?
still not really a very good working medication that people can take. There is also a lot of things going on about gene therapy, because most of the sodium channel blockers or medication that you could take is not specific for a single sodium channel. It always works on all the different subtypes, and we have few different in the brain.
And that leads us back to the original statement you made before, where if you're targeting one channel, but you're medicating that channel, but all the channels are affected, you have to weigh the consequences of those side effects. Yeah.
Exactly.
Gosh, that's a hard decision for parents because you, of course, want to medicate your child so that the symptoms stop, but you don't want to cause more harm. That's just it's difficult choice, but you have to rely on your physicians and researchers. When you look at research that you've done in the past on any of this, what's something that was like, you're so glad you researched this because you actually discovered this really good, it really helped advance what we know?
I mean, I just can come back to the KCNA2 story, because this was really, I mean, this was the most translational story that I had. That something that we found out in the lab went into the clinic, and also quite fast. And this was just able because the 4AP was already used in the clinics. So it was a repurposed drug. It was
Good.
already used for other diseases, and it could just be given to the patients.
Right. When you say that, my own child's on 4AP, so I can validate everything you're saying because we had to go to the hospital, get titrated up, make sure the side effects were not, severe, make sure she could tolerate it, and she has done better in so many developmental ways after being administered 4AP. And my assumption is you have to stay on it. It's not like you're taking it and your brain learns around it and you can just come off of it. I don't know. What is your thoughts on that?
Mm. That's for me, it's difficult. So I think that it's if the patients are doing good and they tolerate everything, then I think you just should stay on it. I mean, you can also try at one point to just get off the drug, but in the patients that I know, at least the seizures came back and the kids were worse again. So, but of course, this is all. I mean, these findings are also important, that we get to know if it's really working nicely. So to get off a drug and see that it's worsening, and then get on it again and see that it's getting better, that's also important information for clinicians and for researchers.
Exactly, because I was approached by a few researchers asking if I could do that, decrease it, see what happens, but I'm too afraid as a parent. But I know other people have done that, and then they've gotten back on it. So that does give us information that it's working, but
Yes,
it doesn't work for everybody because even if you have the same diagnosis on paper, everybody's different because there's so
of
many other
course.
variables in your body that are working for and against you, either way. What is your next steps? Like, what's some of the next research you'd like to do? Like, if you were given $10 million, so money is not an option, or not an option, not an obstacle, what kinds of research would you want to see next?
I mean, there are so many possibilities now that we also have genetic testing, so we could do really cool things also with our mouse models, do sequencing of the mice and to see which other channels or other proteins in our body are up or down regulated that are also in or could be also involved in the disease. So there are many things that I would love to do. So sequencing, I mean, $10 million would be a lot. I could buy so many
I know.
things for the lab, new microscopes, get best people here to do research. Yeah, but I mean, would still look on the path mechanisms, so the mechanisms that are that are causing the disease or different diseases, and to find out new mechanisms and get therefore new treatment options. So this is
That
still my goal, and therefore we are in the lab to
that
find
makes sense.
out.
I mean, if we could learn more about what's going on, we could
Exactly.
maybe figure out, okay, what can we do about it, you know?
Exactly.
And the one thing I just from everybody, I will speak for all the parents. We appreciate everything you do in that lab because it matters to us. We live with this 24/7. We watch our children thrive, but we also watch all of their challenges. And it's sometimes very, very, very difficult.
I
Not to
can
mention we
imagine.
lose some of these kids, and it's unthinkable. I can't even explain, how what it is to like to talk to a parent who have lost a child after all the hope that you've had. Um, I guess I have one last question for you before we close today. What is something that you would tell new researchers, like people that are just getting into the field and they're not familiar with rare epilepsy? How would you encourage people to take a look at this field and join your group and join this endeavor?
I would tell them that that this is really necessary because not many people, especially not the big industry, care about those rare cases, and that it's really important that that research is going on and that we really look what is going on in these diseases, in these channelopathies also. I mean, there are also other rare diseases, but it's very important that there are people working on this um, to help those people.
Yeah, and really, like, it's kind of exciting. They're kind of in the beginning of something. So if you were a new researcher and you started your career in this lane, you could end up discovering things and really getting to know a small group of researchers and make a huge difference throughout your career. That's what I keep thinking about it. Um,
It's not just that we find out things about the disease. We also find out, yeah, or we learn things also about the brain and how things are working there. So it's not just that we are, I mean, this is the main thing that we would like to help people, but we also find out new things, how our brain is working actually.
and that can help. That information can be generalized into many different
Exactly.
avenues. That or areas. That's great. Um, well, Dr. Uli, thank you so much for today. I'm sure that people listening are grateful for this information and they've learned a lot. We also have a glossary, so any of the terms that we use during this podcast, you can look up on the glossary to redefine them and read them again. Also, if you want more information about KCNA2 epilepsy, you can go to www.kcna2epilepsy.org. So thank you so much. I really appreciate your time today.
Thank you, Nancy.
Thanks for listening. If you enjoyed this episode, be sure to subscribe and share this podcast so that more people find us and we can keep the conversation going.